Genetic determinants for simple interactions lay the groundwork for more complex interactions to evolve. Evolutionary theory must account for the origins of the genetic apparatus from simpler forebears. Contrary to the pseudoscientific belief in irreducible complexity, concrete empirical studies show that complex structures and functions do not arise suddenly. Complex functions do not arise because of a single opportunity or selective pressure in the struggle for existence. Rather, they are blindly cobbled together by what might be described metaphorically as Rube Goldberg machines, tinkering, opportunism, or trial and error. This is metaphorical because there is no intelligent design involved. Natural selection acts, as Richard Dawkins aptly puts it, as a blind watchmaker. Often, adaptations that arise in one ecological context are reused for different purposes in different contexts, a process known as exaptation. With this in mind, can we imagine some hypothetical early steps in the evolution of the enforced correspondence of amino acids with particular codons in the genetic apparatus? If the genetic apparatus gradually evolved from the coalescence of simpler units of structure and function, what are some of those simpler units, and how could their emergence be useful in the struggle for existence at the origin of life? Useful molecular structures and functions in the hydrothermal mounds. The following is a list of the sorts of structures and functions that could play useful roles in capture of energy and nutrients and dispersal throughout primitive hydrothermal mounds. The hypothetical structures and functions are listed in an arbitrary order, and it is assumed that these structures and functions evolve in parallel as initially independent units. As these independent units arise, they have EEP that leads to their coalescence into higher levels of structure and function, gradually leading to a primitive and then a more refined genetic apparatus by natural selection acting at the molecular level. 1. Synthesis of amino acids. 2. Linkages between amino acids and other organic compounds. 3. Synthesis of compounds with high-energy phosphate bonds. 4. Synthesis of ribonucleotides and ribonucleotide triphosphates. 5. Synthesis of peptide bonds, oligopeptides, and polypeptides. 6. Synthesis of polyribonucleotide sequences, RNA. 7. Coupling of inorganic catalysts to organic compounds. 8. Formation of organic aggregates with complementary functions. 9. Tethering of organic molecules or aggregates to porous tubes. 10. Release of organic molecules or aggregates from porous tubes. 11. Dispersal of organic molecules or aggregates in hydrothermal mounds. 12. Formation of RNA chains with catalytic capabilities. 13. Formation of secondary structure and base pairing in RNA sequences. 14. Synthesis of polymers of sugars such as glucose, primitive carbohydrates. 15. Synthesis of glycerol and fatty acid chains, primitive lipids. 16. Synthesis of short and long peptides. 17. Synthesis of polypeptides with secondary structures that bind to other inorganic or organic structures. 18. Diversification of side chains of amino acids. 19. Synthesis of long polypeptides with inorganic catalytic cofactors. 20. Synthesis of long polypeptides with organic catalytic capabilities. 21. Folding of long polypeptides into 3D shapes, tertiary structure. 22. Binding of complementary 3D polypeptides into aggregates with quaternary structure. 23. RNA and/or protein aggregates that bind specifically to smaller RNA molecules and specific amino acids and link them together with strong chemical bonds, leading to primitive tRNA and primitive forms of AARS. 24. Refinement of primitive AARS specificity as amino acid side chains and primitive tRNA diversify. 25. Synthesis of simple RNA templates that function as primitive codons. 26. Primitive ribosomal functions. 27. Refinement and coalescence of primitive ribosomes, codons, and other molecules in evolution of the genetic apparatus. 28. Evolution of DNA as the complementary storage genetic material. 29. Synthesis of primitive mRNA.